Alright, so this next section is going to focus on the nucleus, DNA, RNA, and some of the cellular processes that we're going to talk about in this class. So we just went through some of the major organelles and talked about their function in the cell. So we'll move on to the nucleus. The nucleus stores DNA. There's a few parts to the nucleus that you need to know. First, the nuclear pore. The nuclear pore allows for substances to move into and out of the nucleus. The nuclear pore is found in the nuclear envelope. So the nuclear envelope is the boundary of the nucleus. These nuclear pores are highly specific, so they're selectively permeable, and they're much more selectively permeable than the cell membrane. So we have our cell membrane, we have our nucleus. The substances that want to get into the nucleus first have to make it through the selectively permeable cell membrane and then have to get into the even more selectively permeable nucleus. So if we have drugs that we're trying to alter our DNA function with, they have to get through all of these layers in order to actually access our genetic material. And that's good because we want our genetic material protected. This genetic material is all of the directions that make us an individual. If we have damage to any part of this genetic material, we could have cell death or cell mutation, and cell mutation could lead to the development of cancer. So we want to make sure that this DNA is heavily protected. So we have these nuclear envelopes that encapsulate the nucleus and prevent certain substances from getting into to have access to our genetic material. The nuclear pores are really the only way that substances can get into and out of the nucleus. And then we have chromatin. Chromatin is condensed DNA. And then we have this nucleolus. The nucleolus is this really dense region, and that's where ribosomes assemble. So the nucleolus is compacted and really dense chromatin or condensed DNA, and ribosomes are made or assembled within the nucleolus. We'll talk a little bit more about what ribosomes do later on. So since we just brought up DNA, we have to talk a little bit about the structure and function of DNA. Now we already said that DNA is made up of nucleic acids. And nucleic acids have three basic parts: the sugar, the phosphate, and the base. And we said that DNA is double-stranded. So I'm actually going to pull up a second slide so that I can draw this out a little bit easier. So when we have our sugar and phosphates, this makes up the backbone of one of the strands of DNA. But we have two strands to our DNA. And we have bases that line up in the middle, and they're going to bond together. And I showed you that picture in last week's slide of the different bases that are bonded together. We have adenine, which is A, thymine, which is T, guanine, G, and cytosine, which is C, and these two form bonds and these two form bonds. So if we have an A on this side, we would have to have a T on this side. And if you remember, 2 hydrogen bonds form between A and T, so those are hydrogen bonds that are going to hold these 2 strands together. Now if we had a G on this side, we would have to have a C on this side, and there's 3 hydrogen bonds that hold those together. And if we do a T on this side, we have an A on this side, and again 2 hydrogen bonds that hold those together. So this is the basic structure of DNA. Now if we look at this picture, we see that DNA is actually twisted into an alpha helix structure. Here you can see the bases in the center. The backbone consists of alternating sugar and phosphate groups, and then these hydrogen bonds hold those strands together. So DNA is double-stranded, and it's an alpha helix. Those double strands are known as being anti-parallel. And that means that the strands run in opposite directions. We have different end groups that we put on here. There's a 5' group and there's a 3' group. Now because they run in opposite directions, I drew the pentagons pointing up on this side, but the pentagons pointing down on this side, because we have opposite groups attached here. Now this strand runs in the 3' to 5' direction. This is going to be important when I start talking about DNA replication. But now you're familiar with the basic structure. You've got this backbone that consists of alternating sugar and phosphate groups, and then you have the bases that line up in the center. A pairs with T, G pairs with C, and the hydrogen bonds hold those strands together. So what does DNA really do for us? Well, DNA is a sequence of bases, and those base sequences are going to be turned into a sequence of amino acids. And that sequence of amino acids gets turned into a protein. And the proteins not only physically make up our body, but the proteins can serve as enzymes that are going to do certain functions inside of the body. And those enzymes dictate everything that happens to us, what we look like, all of our senses, all of our interactions with the environment, our responses to the environment. It's all dependent on what's encoded in our DNA. There's a couple different things that are going to happen with our DNA. We can either make an entire copy of our DNA, or we can use our DNA in order to make a protein. So I'm going to talk about both scenarios. Let's say we have a cell. And inside of our cell we have a nucleus. And inside of that nucleus we have 46 chromosomes. And chromosomes are just small segments of DNA pieces. Humans get 23 chromosomes from their mom and 23 chromosomes from their dad. And those pairs of chromosomes are inherited. So they came from our parents. So every individual in every one of your cells, you've got 46 chromosomes. The only cells in the body that don't have 46 chromosomes are your blood cells, because they don't even have nuclei, like your red blood cells, and your gametes. So your sperm or your egg cells, they don't have 46, they only have 23. But every other cell in the body has 46. Now let's say we want to make a copy of this cell. We want to go through cell division. If we just split it in half, each cell would only have 23 chromosomes. We don't want that to happen. We want it so that we can make identical copies of our cells. If you get a paper cut on your skin, you want to be able to regenerate those cells and grow new cells to repair that paper cut. So instead of just taking the 46 and splitting it in half, we actually have to double the genetic material so that we have 92 chromosomes. And then when we split them in half, we'll end up with equal cells that have 46 and 46. So how do we double the chromosomes? Well, we have to copy all of the genetic material inside of the nucleus. This is done through the process of DNA replication. So DNA replication is talking about making a complete copy of all of the genetic material inside of your cell. I'm just going to go through the basic steps of DNA replication, just so you get an idea of how it works. So you have this double-stranded DNA here, and I'm going to draw it on its side. The bases are all lined up in the middle. So now we want to make a copy of this genetic material. We need to access what's on the inside, because eventually what we're going to want to do is we're going to want to separate the strands from each other. And we're going to want to build new DNA on the inside so that we end up with identical copies. So we use the existing strand as a template to make the new DNA. This is known as semi-conservative replication. So you're going to use the existing strand as a template to make the new strands. So again, how do we do this? So the first thing that has to happen in our strand of DNA is we're going to have to open up the strand. So if we have our DNA that looks like this, we have to have an enzyme that comes in here and separates those strands, because we're going to want to push them apart so that we can build new DNA on each of the insides of the strands. That enzyme is known as helicase. So what helicase is going to do, it's going to come in here and open up the strands of DNA. The next thing that's going to happen is we have to lay down a primer. Now a primer is made up of RNA because we can't just make DNA out of nowhere. That's one of the limitations of making DNA. It can't just arise out of nothing. That's called de novo DNA synthesis. That can't happen. We have to first lay down a piece of RNA, and then we can eventually lay down the DNA. So we have to put down an RNA primer. So the next step is RNA primase is going to lay down an RNA primer. And DNA can only be replicated in a particular direction. So since our strands run in the 3' to 5' direction on the top, and they run in opposite directions on the bottom, we actually have to synthesize this strand in this direction, and the bottom strand has to be made backwards. So we're going to lay our RNA primer in this direction. Then the next step is we have DNA replication happening. So DNA polymerase III, or in the case of people, there's a bunch of different types of DNA polymerases, but DNA polymerase is going to start replicating the DNA. So now we're going to lay down new DNA here. So that looks like a mess so far, but it's about to get worse before it gets better. So helicase is going to continue to open up the strand of DNA. And as our strand continues to open, we're going to lay down one primer again for the top strand. We laid down one primer for the bottom strand. We replicated our genetic material, and since the top strand is going in the same direction that helicase travels, the top strand is synthesized continuously, so everything's good with that one. The bottom strand is an issue. Since the bottom strand has to be synthesized backwards, now we have to lay down a new primer when the next section of DNA opens up, and a new chunk of DNA is made. And these chunks of DNA are known as Okazaki fragments. So we have the leading strand on the top, and that leading strand is going to be synthesized continuously because it travels in the same direction that helicase is opening up the DNA. The lagging strand is made backwards in chunks, and those chunks are called Okazaki fragments. And that's a problem. So when we continue on here, and I'm going to add another one of these, in the end, our DNA is going to look something like this, where we have the original template strand, and then we have one primer on the lagging strand, or leading strand, and we have multiple primers on the lagging strand. This strand is going to be made in nice one continuous piece, while this strand down here has all of these Okazaki fragments. So the next thing that has to happen once DNA has been replicated is RNA primase has to remove the primers. So any place that you have a piece of RNA, it's going to be removed by RNA primase. Then replaced with DNA. So that looks a little bit better, but we still have an issue. There's still these gaps that exist between these bases from the Okazaki fragments. So DNA polymerase 1, replaces RNA primers with DNA. And then the very last step, we have this ligase that comes and glues the Okazaki fragments together. So here you'll have this connection made and this connection made, and now they look identical. Now you've got two identical copies of DNA, and each one can go to each cell now. So that's the basis of DNA replication. You may go into more detail once you get to advanced A&P. Certainly in microbiology, we review this process, but you need to understand what's the underlying purpose of DNA replication for this class. DNA replication is the process of making a complete copy of DNA, and we need to do this in order to have cell division happen. If we want to replace damaged cells, like skin cells that have been damaged, and we need to make more copies of them, we want to make identical copies. So first we photocopy all of the genetic material, and then we split it in half into separate cells. DNA replication is just the process of making a complete copy of all of the DNA inside of a cell. Now, before we go on to talk about protein production, we should talk a little bit about the difference between DNA and RNA. So first of all, the name, deoxyribonucleic acid versus ribonucleic acid. And it's named different because there's a different type of sugar found. So instead of deoxyribose for DNA, it's ribose for RNA. The bases are also different, and we talked a little bit about this, but the bases for DNA are A, G, C, and T, while the bases for RNA are A, G, C, and U for uracil. DNA is typically double-stranded, while RNA is typically single-stranded, although there are some very important differences and exceptions to this, especially when we're talking about viral genomes in microbiology later on. But double-stranded for DNA, RNA is typically single-stranded. DNA has this big kind of storage capacity. So encoded in the sequence of bases are all of the directions that make an individual. Now, DNA codes for proteins, and stores all of those directions that make an individual, while RNA is more directly involved in making proteins. As far as the types of DNA go, you have the genomic DNA, which stores all of the directions in the nucleus. But you also have mitochondrial DNA, and that mitochondrial DNA has now been linked to many very important diseases. So it has a very huge impact on human health. That mitochondrial DNA, we'll talk a little bit more about in microbiology as well as potentially in advanced A&P. But mitochondrial DNA, you only get it from your mom because the egg is the gamete that has all of those organelles. So the sperm really just delivers genetic material, while the egg has all of the organelles from the mom. So you only get your mitochondrial DNA from your mom, but it does have some association with disease, which we're not going to get too much into in this class. Well, RNA, there's some different types. There's rRNA, which is ribosomal. That's what makes up your ribosomes. So your ribosomes, which we'll talk about next, that's just a bunch of folded pieces of protein and RNA. You've got mRNA, which is messenger RNA, and tRNA, which is transfer RNA. And that's something that we'll talk about in just a little bit, like what the purpose of those different types of RNAs are. So you know ribosomal makes up the ribosome. We'll talk about messenger and transfer RNA in just a minute. So that's basically it. Other than that, they're both alternating sugar phosphates. They both got the bases. You can find DNA specifically in the nucleus, while RNA kind of floats between the nucleus and select organelles. Major functions, DNA is the major storage of all of the genetic information, while RNA It's usually smaller and just aids in protein production. And then there's some derivatives, some other types of RNAs that are involved in regulating the cell, which we're not going to talk about in this class. So the ribosome. The ribosome is responsible for protein production. So the ribosome is where proteins are made. You have free and attached ribosomes. Free ribosomes are floating in the cytosol, and they're going to make proteins for inside of the cell. Attached ribosomes are going to be on the rough endoplasmic reticulum, and these are going to make proteins for secretion or insertion into the cell membrane. So attached ribosomes make proteins that are destined for the outside of the cell or the cell membrane itself.